Hello, my name is Vipul Upadhyay and I am recording the stage 2 presentation of the WinBio 2022 internship. So the topic of this stage was related to molecular dynamics, MD simulation, basic Linux and SSH secure shell. So I covered the topics and I belong to the group MDS. The content you can see here, it will start from the basics of docking, types of docking, overview of the steps. Then I will talk a little about AutoDock and AutoDock vina. Then I will show the steps of preparation, then few demonstration by taking the protein and ligand. Then we'll move to the basics of MD simulation and GROMAX. Then we'll see the demonstration of the MD simulation of the lysosome in the lysozyme in the bottle. So when we say docking, it basically means we are trying to figure out the basic configuration, basic orientation of any stable interaction between protein receptor, protein ligand. It can be any two molecules. Generally, we do the docking for protein and ligand or any small molecule. So the basic goal is to know the interaction between the two molecules that we have taken. Is it favorable or not? If the interaction is favorable, if we get the yes, then we find what is the orientation and we try to analyze what is the stable complex structure. So in this it includes two minimum major things, which is finding the minimum energy of the stable complex and the maximum interaction that could be possible. So interaction could include various bonds like hydrogen bonds, hydrophobic interactions, ionic, etc. So when we talk about docking, taking the two molecules like protein and ligand, there is a concept of active sites. So these molecules take interaction take place at some specific points. So it can be a binding site or catalytic site. So if it is a binding site, it is binding to that particular position. It is either activating or inhibiting. And if it is an enzyme, then it will be a catalytic site. So it will be performing some specific task. So as I said, there could be inhibition or activation. Then there are many proposed theories for enzyme action models. Yes, these are some of them. So lock and queue, induced fit, and transition state. So when we are taking the docking into consideration, we can see various degree of freedom of a molecule and the interactions. So here is the basic overview. We take the protein or receptor structure from PDB, or we predict like here it has in alpha fold, or we make our own architecture also. So the second is to take the structure of ligand through PubChem or Zinc databases. Then we do the molecule preparation. So these are the basic steps for before performing the docking. Then we try to find the binding site prediction tools. If we know the binding site, then it is good. If no, then we use some tools to predict them. Then we perform the docking. So we can do protein, we can do other protein ligand, protein small molecule also. So here we perform through AutoDock or some other techniques. So in this tutorial and this presentation, I will be performing through AutoDock. So then we visualize the molecule through various visualization tools like PyMol, VMD or RASMol. So these are the types of docking. Basically, I have told you it could be protein. It could be protein nucleic acid. It could be small molecule or nucleic acid. It could be small molecule or protein. So majorly we are doing small molecule and protein, or in some cases protein and protein. So it can be divided into various types based on binding site. It could be blind docking, targeted docking. Blind docking is taking whole receptor molecule uh, as a grid to perform the docking. Targeted could be related to a specific position that we know. So basis of molecule I have told protein peptide, protein ligand, and we can also do the protein like DNA. Then on the basis of flexibility, it could be rigid or flexible. So receptor could be rigid, ligand could be flexible. Receptor could be flexible, ligand could be flexible. So as we increase the degree of freedom, as we increase the flexibility, the complexity of the computation also increases. Then we can see the overview steps of docking. So take the protein, search in PDB, find the structure. Same for ligand, do the docking. So protein and ligand structures are prepared. They are matched to form a perfect conformations. So these conformations are called poses. So poses are ranked according to the number of the least energy and also the interactions. So here is the basic overview of AutoDock and AutoDock vina. So these both are molecular docking programs to predict the binding of small molecules and protein. So AutoDock vina was developed and it is a newer version and faster and efficient version. So it's based on the global and efficient optimization. So two components are present in the AutoDock vina. First is AutoDock tool, which is used for preparation of molecule, then vina, which actually performs the calculation. So these three are the basic input files that vina takes: processed receptor, processed ligand, and configuration file. Now you'll see basic protein-ligand docking protocol. So I will not explain this in much detail. I have already talked about this, taking the protein and making visualizing through basic PyMol or Chimera. I used Chimera. So removing some non-standard molecules, then removing uh, hydrogen molecules, then preparing grid. So grid preparation is very important because that is the area that we are specifying to perform the docking. Then ligand preparation, same. Then we do the analysis. So this is the basic overview of, again, I will not repeat. I have discussed this many times, that these are the basic steps of preparation of receptor and preparation of ligand. So this is the first demonstration. I took the receptor 7OGJ. This is basically a tmRNA transferase. You can see here, this is a transferase protein. The ligand was SAH, which is in complex with this. So I used camera and AutoDock vina. So I used several ways. So the first part is without energy minimization. So I take these two receptor and ligand, and I perform the docking without any energy minimization. So this was the result I get, and you can see here in the chimera. This is the interaction. This is the molecular view. Then the second part was to perform the docking following the energy minimization, adding hydrogens and charges and etc. docking preparation. So I did these preparation. You can see here in the left panel. So these are energy minimization, then the docking preparation, which includes addition of hydrogen and charges, and also the amber-based standard residues charges are applied. So this is the basic structure. You can see this is the protein only. So I have removed the non-standard hydrogen if there was present or any other molecules. Then this is was the result I got. So after performing the energy minimization and docking preparation, there was a slight change in the result. So this is the result. Uh, you can see the scores and RMSD lower bound and RMSD upper bound. So this is the hydrogen bond analysis. So I also did the hydrogen bond analysis. So you can see there are the number of hydrogen bonds is listed in front of the scores. So these are the hydrogen bonds even depicted in the picture, and this is the surface density structure of the receptor plus ligand. This is the demonstration too. I took another protein, that is BDNF. Growth factor receptor is important in neurodegenerative diseases. I performed the same as I have told, chimera and AutoDock vina. I took the whole complex. So you can see some non-standard atoms here. So this is the whole complex downloaded from PDB. Then I did the preparation. So this is the protein I removed those atoms, and this is the basic docking preparation that I've told you. Then this in this I also did preparation for ligand. This was one extra thing I did in this uh, demonstration. So this is adding charges, gastrical charges to the ligand. Then I performed the then AutoDock vina, and I took this grid. You can see the grid preparation here. Then this is the result. So this is the basic scores and RMSD lower bound, upper bound, and this is the hydrogen bond analysis of the same. So this is the enlarged view of the molecule. So you can see the hydrogen bonds here. So this is the hydrogen bond which is made between ligand and the receptor. So this is demonstration 3. I used complex and the basically I did ADF used ADFR scripts and command prompt in this. So instead of using Chimera integrated with AutoDock vina, I used Chimera for preparation, but for performing vina, I used command prompt and ADFR scripts. So this is complex. I took the same molecule. So I prepared configuration file. So by using Chimera, I prepared this configuration file. Then preparation of receptor and ligand using the ADFR script. Then this is the result. Total 9 poses. So affinity of all the poses is given and RMSD value. So you can see there is a better performance when I used the AutoDock vina executable and did the preparation through ADFR scripts. Then this is the basics of MD simulation. So MD simulation includes, so here as you can see, step 1, building the simulation box. So again it starts with taking the PDB structures, then uh, building a box, then do energy minimization, then some boundary conditions, then equilibration, NPT, NVT. So these are the basic things that happens, and then we do the main production run. Then we do the analysis like RMSD, RMSF, hydrogen bond, etc. So GROMAX is the basically MD simulation software package that is used for basic simulation of biomolecules. So this is an open source software, and it runs on can be run on many operating systems. I use Linux. So this is the overview of the GROMACS using GROMACS software. So we take the PDB. So if ligand is present, then we have to remove the ligand. If not, then we move to PDB to Gmax. So it generates topology file and etc. Force field conversion, etc. Then we do the solvation. Then we do neutralization by adding ion through J ion. Then we do the energy minimization, and then we do the equilibration. So NVT and NPT equilibration. Then we perform the basic MD run. After MD run, we do the analysis. So this is demonstration of lysosome in water. So these are the basic steps, and this is one is the lysozyme. One PDB ID is one AKI. So taking the PDB structure, then applying GROMACS module PDB to Gmax, then selecting the force field. So we take the cubic box for our edit conf, then we add water, then we add ions, and then we generate the TPR file. Then we neutralize by adding charges, and then we do the energy minimization of the whole ensemble. Then we do the equilibration. So it takes the constant number of molecules, constant number of temperature, and the volume. Then it takes NPT takes the number, pressure, and temperature. Then we did the production MD run for 1 nanoseconds using MD run. So this is the analysis after performing the MD run. So this is the RMSD value. This is the radius of gyration. So this is my bibliography. Thank you.